Okay, so we're going to discuss today here, okay, the gram staining reaction. So gram staining. So gram staining has been introduced by Hans Christian Grams in 1884. So this is a type of the staining reaction that would eventually help us then to differentiate our bacteria into your gram-positive and a gram-negative organisms. So gram-positive organism here are those organisms wherein they retain the color of the primary stain, which is the crystal violet, making them here as appearing to be color blue or black or violet color. On the other hand, the gram-negative organism, on the other hand, they try to retain the color of the counter stain, the safranin, that would enable them to be colored here as red or pink na color. Sometimes we're able to encounter organisms which are gram variable. Gram variable organism are actually gram positive na organism, but after staining of the gram stain here, they tend to appear to be gram negative. Usually the reason for that, it might be because of the age culture. So medyo matagal ng culture natin, hindi siya ganun ka fresh na culture. Or most likely, the gram variable appears to be like belonging to the spirochetes na mga bacteria natin. Those are mga spiral na bacteria. Okay, so the gram staining reaction here is based on the stain uptake of our gram-positive to gram-negative organisms because of their differences in their cell wall structure. So gram-positive would have your thick na peptidoglycan layer, whilst your gram-negative would have your the cell wall made up of the thin na inner peptidoglycan layer, and we have thick na outer membrane is primarily made up of the lipids. Okay, so the primary stain used here would be your crystal violet. Then we are also adding bicarbonate, merthiolate here. Bicarbonate would enhance the colorization of your gram-positive that would enable them to be colored as blue. And merthiolate here is being added to prevent also here the contamination with sodium bicarbonate with a small gram-negative bacilli producing a false positive. So, hindi sila magiging blue na color ang gram-negative natin. So, we're also adding here grams iodine as or mordant. So, pag sinabing mordant, it helps your stain to be taken up by your bacterial cell. And we have also here your decolorizer in the form of your acetone. And another one, we have your counter stain. We have here the safranin. So basically, again, ang nangyari dito, once we try to add here your primary stain, that will be taken up here both by your gram-positive and a gram-negative na bacterial cell wall. And apparently, with again able to take up your violet here for both gram-positive, gram-negative by addition also of your bicarbonate merthiolate complex plus your mordant, your grams iodine. That will enable your bacterial cell here to take up your primary stain. Okay, then upon addition of your decolorizer, your acetone, if that one is your gram-positive, because remember gram-positive would have thick na peptidoglycan layer, which is much of the protein structure, it would enable the cell here to form a coagulum. So pag nag-coagulate siya, eventually that would try to trap here your crystal violet within the cell wall. In the case of your gram-negative, so remember that your gram-negative cell was primarily made up of the much thicker na layer ng lipids. So upon addition of your acetone, it will form here pores. Okay, so eventually what's going to happen here, your crystal violet will going to escape. So therefore, upon addition of your counterstain the safranin. So, since for the case of your gram-positive, since na nag-coagulate na siya ang kanyang cell membrane, and try to trap here your primer stain, your counterstain will not able to penetrate. Therefore, your gram-positive organism here try to retain the color of your primer stain, the crystal violet, making your gram-positive here as colored blue or violet in color. Whereas for the gram-negative, after addition of your acetone, since your crystal violet has already escaped, so upon addition of your counter stain to safranin able to penetrate the safranin, and therefore they're able to retain the color of the counter stain to safranin, which is red na color. And therefore, your gram-negative would appear to be pink or red na color. So, color ng safranin natin almost pa siyang orange-red na color. Okay, then we have here the procedure for the gram staining. So first, you're going to make your film just like what we have performed in the preparation for smear. So from your biological specimen, again, if it was solid, then lalagay tayo ng saline solution. Then if it one coming from a broth culture media, so you don't need to add your saline solution. So try to make here a thin smear of that. Okay, then heat fix by allowing your slide here to pass through the under the flame for several minutes. So do not over flame that one to prevent the disruption of your bacterial cell. Next, you cover here with your safranin by flooding. So magdadrop ng crystal violet, I mean, that would allow here the entire surface of your smear here to be covered by your primary stain. Then you add 3 to 8 drops of your sodium bicarbonate methylate. Try to rock your slide gently. So rock mo, ganun lang siya. Parang ganun. Okay, gently for 5 to 10 seconds. Then you add here your grams iodine. So ipa-flood mo siya ulit ng grams iodine that would replace your crystal violet. Okay, then try to rock gently again your slide for 5 to 10 seconds. So after that one, okay, you try to hold your slide vertical position, ganun. Or pwede yung ganyan, slightly tilt. Okay, try to add here acetone. So dito ka sa taas, mag-add ng acetone. So magpa-flow ang acetone natin dito. Okay, that would allows the decolorization or the washing off of your crystal violet. Okay, then air dry your slide. And eventually, finally, you cover the slide again here with a counter stain, your safranin. So, i-cover mo siya ulit. Okay, and okay, try to wash off. Try to rock gently. After covering with the safranin, i-rock gently ulit natin and try to for 5 to 10 seconds. Then after that one, okay, try to wash off your slide here with your tap water. And then blotted it dry. Okay, so notice here na hindi tayo nag-wash off ng water in between of our application of the stain. It's because here pag nag-add ka ng primer stain, hindi tayo nag-wash off ng water. Okay, but we tend to have continuously adding the stain here from your primer stain next with your sodium bicarbonate methylate, then you add also your grams iodine. So hindi tayo nag-add or nag-wash off ng water in between because there will have here baka magkaroon ng unintentional decolorization of your organisms here and therefore magkakaroon siya ng ibang reaction. So baka instead of the gram positive, nagiging gram negative siya. So therefore, nasa last step na ang ating washing of the water for that. Okay, and after that one, air dry mo smear and then try to examine the morphology of your bacteria, noting here if what is present would be gram positive ba or gram negative. At the same time, you should um, also observe here for the morphology or the arrangement. Is that in cocci? Is that in bacilli? Also, is that in pairs? Is that in chain? Something like that. This also some of the things that you need to write in your gram stain results. So parang like gram positive, cocci, in pairs, in chain. So ganun ang reporting natin. Or gram negative, bacilli, diplobacilli, something like that. Okay, dry examine that one until the OIO because this one is a dry smear. Okay, so thank you.